Okay. So, hi everybody. I'm Yojana Gadiya from University of Bonn. I'm a PhD student, and today I'm going to talk about patent landscaping. So, we all know in drug discovery, there are a number of ways in which you can have data. Most prominently, these data, we aggregate all of these different data into a knowledge graph or a database. If you don't know what a knowledge graph is, it's similar to a figure which is shown on the right, where you have different connections between genes, diseases, side effects, and so on. But when we are collecting all of this data, we mainly look at certain specific data. For example, when we are looking at data from the public domain, we look at data that is coming from publications, we look at data that is coming from experimental validations, we look at, to some extent, from clinical data, and want to add more provenance to this data, you would add in your internal or in-house data, which would include internal experiments or internal manual curation pipelines. But in either of these cases, what you're doing is you're just aggregating data that is publicly available and has been mostly researched by academicians. Something that we are totally missing in all of this aspect is patent data, which is being used mostly by pharmaceutical companies, for example, for promoting their research and development. I talk about patent data, but what are patent data? So particularly patent document is basically a document that discloses kind of a novel. Okay. So a patent document is basically a document that discloses a novel invention to the public. And to do so, you have many interests. So you do this so that you have a legal protection for your invention. And second, it grants market exclusivity rights. That means the patent owner has the sole right to produce and sell this particular invention out. To register these patents, you usually go to specific designated patent offices that are all around the globe. So they can be in States, they can be in Europe, and so on. And when you submit these patents to the patent officer, what they do is basically the patent officer is going to read your document and start to summarize the context of these documents using certain codes, which are called as the International Patent Classification Codes. And these codes, you can see that these are kind of a hierarchical codes that talk about a variety of domain, because these patents are just not for drug discovery. They can go for everything, including your shoes, your clothes, the glassware, as well as for drugs. I have a poster that describes more about all of this, and if you're interested, you can take a look at that. But one key thing is that these patent documents have been existing for so long, and why hasn't anyone mined them? And there are a number of reasons for this. First, that these are legal documents, so it becomes difficult for someone who's non-scientific or is not part of this field to even understand what these documents mean. Second, that these documents usually are unstructured. That means that if you search the net currently, these documents are usually available in PDF documents, which makes them unstructured and non-machine friendly to some extent. And because of these two classifications, or these two obstacles that we have, mostly you need a dedicated patent firm or a dedicated patent lawyer to actually understand what's happening in these contexts. But things changed around. So in 2015 to 2017, many new things came into place. And when people understood that about 70% of the data that is published in patents is not available in any other form, they started to look on how to make this data more prominent That's where prominent companies like Google came into consideration, saying that, okay, we will create a patent database, which is called the Google Patent, where they will collect all of this data and start to aggregate and structure this data. Similarly, the European sisters for the EMBL and created the Shaw-Campbell database, which tries to restrict the domain of patents to just talk about those that are life science informatics or life science related. But in either of these cases, what they have done is just structured the documents. There is still a bit assistance and still a bit difficulty in trying to access and see what's there in this document. Currently, there are a lot of proprietary tools that help you, such as SciFinder. If you're in the pharma industry, you already have heard of this, that tries and helps you understand the context and mine these patent documents, but nothing is available open source. So what we did is try to develop something in that direction. So that we don't explore and explode all the data resource that is there, we want to restrict to one specific domain to start somewhere. So we start doing with these two indication areas. First, being rare diseases, where we start with OrphaNet, which is a common knowledge graph for rare diseases. And second, we look at Alzheimer's, which is a neurodegenerative disease area. And there's a knowledge graph from the Human Brain Pharmacome project that was already available. From these knowledge graphs, what you can extract is information about compounds and genes. But we want to do it something differently. So we started looking at all the genes that are directly connected to these particular indication areas. And then based on those genes, we wanted to see how we can identify and mine patent documents around those genes. To do so, we couldn't, as I mentioned, that we couldn't find anything that was open source. So we created an open source tool that can exactly do this. It can do this in two ways. The classical way is the compound agnostic style, where you have a compound and you can search for all the patents that are around this compound using public database such as GenCompBio. But if you have a target, what we did is, no worries, you give us your target, we are going to find out all the bioactive compounds that is available in public databases, such as ChemBio for this case. And then now that you have compounds, we can search for patents around this particular target. When we're searching for patents, as I mentioned earlier, that GenCompBio is covering all of life science. But we're not interested in all of life science. We're interested in certain things that are interesting for us as drug discoverers. First, that they talk about drug formulations, for example. Second, they talk about something where the compound is being active or inactive on a particular indication area, or it could be active or inactive with respect to a specific gene. And to do so, we subsetted a class of IPC codes that are actually relevant for this. These are like 16 IPC codes that talk specifically in these indications. And based on these, now you get is a list of patent documents that are talking more about your particular gene or your particular compound. Now that we have patents, what can we do with this? So today I will just demonstrate 3 such use cases from different perspectives. Given that the crowd here is pretty diverse, I will try and hope and manage your expectations with this presentation. The first one being that the patent, when you're looking at patents, you want to look at with respect. Yeah. When you're looking. I'm sorry, the screens on both sides are super different, so I don't know what's happening. I'm trying to figure out. Do you know why these two screens are so different?
To go on full screen?
Yeah, it's on full screen. It suddenly started acting different.
Wait, this is not full screen.
It is full screen.
Yeah. Can we just close them up again? That's your. Do you want to close and open it again? Yep. Okay, it didn't go into presentation.
Yeah, I'm just confused. Oh, it's okay. I can try and navigate if that's okay. Yeah, thank you. Yeah, sorry for that. Okay, so as I was saying that I will try and explain you 3 different case scenarios in which you can extract and understand more about patents. First being patent portfolio. What does that mean? So basically what you do is take all of your patents, respect to one particular patent owner, and align them across annually. So each year you see how many patents a particular company or a particular institute is doing. When you see this, you start to see certain trends. We saw a few that I will highlight here. So the first trend being that when companies are merging, you usually see that there are assets, which are also patents, that are actually being transferred from one company to other. We saw this in the case of Ipsen, as you can see in the highlighted circle there, where you see that in the rare disease field, Ipsen had no patents until 2015, and suddenly there was a spike in the number of patents coming from 2005 to 2007. And when we looked at these patents, they were just, because there were just 5 or 6 of these, we started to see that this was because Ipsen had actually acquired one of the sister firms, which is the Stevix, which was actually working on metabolomic rare diseases, and that's why it had these increasing number of patent documents. The second case is that you see that there are also certain cliffs or certain downhills when you're looking into patent documents. And when you look at this and you think, why would there would be a decrease in the number of patent documents? This actually relates to clinical trials that are going on. So in the case of Takeda, which is the second case that was highlighted, you can see that there's a decrease in the patents just before the pandemic period. And there could be 2 reasons for this. So the first one, because we could track that there was a drug from Takeda that was actually in clinical trials for one of the rare diseases, and it actually failed phase 2. Because of that, they had to withdraw certain patents and reconsider their whole patenting strategy to understand more what's happening. The second, it could be that because it was the initial periods of pandemic, so there were certain places where COVID-19 had already been discovered, and that's why there might be a scene where people have, or industries in this case, have started to focus more on the pandemic side rather than focusing on diseases that already exist. Second perspective. Second perspective. Sorry about that once again. No, not here. Okay, so I start again. Yeah, it's working. Thank you. So you already know everything from here. Let's go here. Okay. So now the second point is that you can do not just for academic and not just for finding out what other industries are doing, you can do something for yourself or for your company. How you can do this is basically look at the different target profiles. So now what we did is for a given indication area, now that we have a list of genes, you can now look at how many number of patents are going for each of these genes and align them with respect to which are the different pharma companies or different institutes that are working on this gene. And one of the heat maps here you see is basically doing that, where we collect all of the number of different patents. So based on how many patents you have, you would see the intensity in the color of color of the map. And here you can already see that there are certain things that highlight. So for example, in this case, you have the CNR2, which is pretty highlighted, and there are a number of multiple pharma companies such as AstraZeneca and Roche and J&J that are working on this target. And because this is an interesting target with respect to neurodegenerative diseases, it's pretty common to see this. But not just seeing it from the investor perspective, you can also see it from a research perspective. Now that you have certain compounds or you have interest in certain targets, you can find out what are the other people also working on these targets and try to foster some kind of a collaboration with them because you have certain ideas. So this kind of a heat map can help you find out certain potential collaboration partners in the same sense. Next, if you have like a list of genes that you're worried about and you don't know where to start from, This kind of a map can help you try and prioritize based on what's happening in the market, because this particular heat map will give you the patent and the market overview of these genes, and you can prioritize and reanalyze your gene list based on those sense. Let's look at another interesting case in the case of rare diseases. In this example, we look at DAO, which is an D-amino acetate oxidase. And this target, as you see in rare diseases, is the only one which has multiple different institutes and research pharma partners being involved in this. And this is merely because this target is interesting in multiple fields. For example, this is basically a neurodegenerative marker for some diseases and also a rare disease. It also plays roles in metabolomic disorders. This makes it a pretty interesting target for multiple different partners or multiple different pharma companies to take a view at this in different contexts. Last but not the least, we can look at, so we are always wondering why not just look at scientific documents and why just look at patent documents. So we tried to do an analysis of seeing the number of publications that you have for a particular gene and compare it with the number of patent applications that happen across the same year in that context. When you look, there are certain patterns. So first pattern, which is pretty common, and what we found was that there is approximately a 4 to 7 year difference between a particular gene picking up an interest with respect to a pharma company or a pharma landscape compared to those that goes into research. Of course, this data is too less to say which one came first, whether it was the pharma company that picked up the target, or whether it was the research that led to the establishment and led to the research of this particular target. So we are still trying to figure out what's going on there, but we at least try to find out that there is some kind of similarity and some kind of advancements that's happening across those lines. Moreover, there are two such cases that I would like to highlight. The first one being that of PYGL, which is a polyglycosyl protein that is present in the body. And this is pretty evident here. You can see that first you see a good amount of peak in purple in that box with respect to patent documents. And then you see a certain steep below in around the year of 2010, for example. And this is probably because that this particular gene was first invented for rare diseases, or first found to be evident in rare diseases. And that's why a number of different partners and number of different research institutes had their interest with this disease and were looking into exploring this particular gene in rare diseases. But as they had explored the whole area, they didn't know what to do next. And that's why the interest on this gene decreased. But then you see that along the same lines, when the decrease in the interest from the patent or from the pharmaceutical side was happening, there was an increased interest with respect to the publication or research side. And this is because there was a classic case of target resurrection, because this target was found to be a major player in diabetes type 2 later, which led to an increase in the number of publications, as you can see. Not all profiles are such opposites, where you have a high number of patent and low number of publications. Other types are more synergistic. For example, with retinoids, RVT, which is a prominent player in insulin and diabetes. And this is a pretty common target where both the patent and the publication profiles match with each other. So, as a matter of fact, I have hopefully, without any disruptions and without you not going haywire along the presentations, I was trying to establish that patent documents are really important. We have tried to create a tool which is called as PEMT, which tries to annotate and extract these patent documents for you. And I have tried to explain how these patent documents can be leveraged for your research based on two cases. First, based on target prioritization, and second, based on how they have been used and how you can modulate those for your marketing perspectives. Lastly, I would like to thank all the co-authors for this paper. All of this data has been published. All of this data is available for you to search. And I thank you for your interruption. Thank you. Thank you for your patience with me while I was figuring out this. And happy to answer any questions.
Yeah, sorry for the technical issues during your talk. We have time for questions.
I have a question about a little bit different use.
Mhm.
How good would it work for extracting information from patterns for plants?
Can you repeat the question? How good it will be?
Work.
Mhm.
So for targets for different sequences which are interesting for plant breeding. So to change a little bit the target which we are looking for, not protein of human, but proteome of, well,
I see.
maize or whatever other.
Got it.
plant.
So currently the pipeline is more oriented towards human, but I think because the code is open source, we can tweak it to include other species because it's basically just querying databases, right? It's querying the Campbell database, and currently the whole pipeline is oriented so that it's least restricted to those in humans. But it's just a changing of a small parameter in that script to run it for any other species. No problem.